Welcome you all to this Cryo-EM course. Last class we discussed about how which one is and which one is a good negative stain image and bad negative stain image. So, in that particular case we discuss about the thick stain and then very light stain, but I did not show you any good images. Before going to good data or good images, it will be better to go through a few more images, because this is you really need lot of practice to see your data and identify is it a good data or is it a bad data. So, lot of time, even if you just remember last class when I was showing the first images where you have lot of white patches and lot of large white molecules here and there, and then some circular molecules. Many times researchers are considering that is their sample, because they are not familiar what is a good sample or bad sample, or what is a good what is a good image and bad images. And that is one of the major reason that you should analyze your data, because from this negative staining experiment you can conclude many things, that you may not get a high resolution 3-dimensional structure, but you can definitely get an idea about the overall morphology of your sample. Even if your protein sample is interacting with antibody, or your protein sample is interacting with the DNA or RNA, that you can also visualize. That I am going to show you during the application part of this, when we will discuss about the application part, how we can apply TEM for imaging of different biological macromolecules. So, that is one of the reason we should go through few more images to identify the good data and the bad data. So, if we visualize more and more TEM images and TEM data, it will really help us in the future to find out that whatever data I am collecting or whatever TEM data I have is we can analyze that data properly. So, now let us take a look the next image or data that whatever we have here. So, in this one you can see that this is our another TEM image. Now, from this TEM image, if you look at and try to find out that okay, I want to see a protein sample or DNA sample or RNA sample, it is quite difficult to identify anything from this kind of images. Yeah, definitely there is a black patch here, there is another black patch here, and some black patches here and here, but if you look at the overall background, there is no difference from the entire TEM image. So, this kind of TEM image, it is called a very light staining images. If you just recall that light staining images again, so if this is your grid and if this is your sample, your staining solution is just staying like that, and here the stain concentration and the thickness is very low. So, this is another example where we are talking about the thin staining sample, and this kind of images are bad images. So, we should not consider this kind of images. So, there is one possibility that whenever you are using the staining solution, probably that staining solution is precipitated, that concentration is not good enough. So, it is a old sample, old staining solution, and it may be possible when you prepare the sample, your concentration of the stain is somehow varied, it is less than 1%, or there is a possibility that when you stain and you blotted the staining solution completely, and that is the reason you are only getting this white background, we cannot see any kind of protein sample. So, this is bad. So, what are the points we should remember? That either staining solution is old. When it is old, what will happen? It will most of the time uranyl acetate and uranyl formate will precipitate, and then concentration of the staining solution will go down. The second option, your staining solution percentage is less than 1%. So when you measure the amount of that uranyl acetate and uranyl formate, there is something wrong, or you blotted most of the staining solution. The another possibility is that glow discharge. So when you did that glow discharge, your glow discharge is not proper, or your grids are not glow discharge at all. So if your grids are not glow discharge at all, and then you are adding your sample and then staining solution, nothing is going to absorb on top of your grid, and then if you are trying to image, you will always end up this kind of bad images. So don't consider this kind of images. If you get this kind of images, it is better to redo the staining again. So when you redo the staining again, you may get much better images. So that time you have to pay attention with all of these 4 points. And what is next? Next we have a different type of images. Here I mentioned that it is a antigen antibody complex. What is that antigen antibody complex? You just consider any antigen, that okay, any viral protein is coming as a trimer, and then we are generating antibody. When this viral protein will injected inside the cell, it will generate the antibody. You find out that antibody, then based on this sequence you can clone it, or you can isolate those antibodies, and then you can make a complex. When you have a complex, and then this complex you can image it. So when you are doing this complex preparation, so what you have there is a 2 ependrop. In this ependrop you have antigen, and other one is you have antibody. You have antibody here. Now what you can do, antibody you will mix it here, and then definitely your concentration it will change, and your volume it will change, and you will get a mix of antigen plus antibody complex. If there is a good affinity, you will get a good amount of complex. Now look at this, that we prepared this sample in this way, and then we are trying to image it. Now when we are imaging this one, we can see there are lots of this kind of protein sample. In this one, those whites are the protein sample here. Now in this particular case, you can see lot of protein sample, and some are like that. So as I mentioned previously, we are imaging some tetrameric protein sample. Now if your antibody it will bind, size it will increase of that trimer, and overall molecular weight it will increase, as well as the shape and size it will increase. So here we are looking at that size at the same magnification, and we are trying to isolate that, do we see any extra density. If you remember first class, when you are talking about that different types of negative staining images, we talk about that trimeric sample, right? Your size will be this, but when a extra antibody it will bind, overall size it will change, and in this sample you can see that overall size is changed lot. So specifically this particular sample, or if you just pay attention this particular sample, if I just remove that circle, it will be clear to you guys. Here you can look at that sample. in this particular sample, where you can see overall size at the center, there is some center of the sample there are a trimeric species like this one. Here if you just look at the center, something it is equivalent to this molecule, and then there are some extra densities are attached here. Those may be the antibody or Fab complex. So here there are many are like that. One is this one, one is this one, one is this one. And also you can distinguish this protein sample from the background. If this is your background is very nicely separated from your protein sample. So one way this is an good image you can consider, because from the background you can isolate your protein sample or your biological macromolecules easily. In that respect, definitely this is an good image. Now come back to the sample part. Now if you look at this sample, there are many places, there are some small particles, so something like that, which are not very familiar with this kind of trimeric sample, so something like this one. So in this case, if I just color it in a different way, though that will be much more clear. So if you just consider, there are many small things are there. So some small things are there. Those are isolated things. They didn't bind with anyone, right? And they are in the background. All are in the background. So that is basically giving us some kind of wrong information. Either this protein is, after forming a complex, there is some kind of dissociation, and that's why we are getting this kind of small species. Another possibility is that if you look at our sample preparation process, what we actually did, we took these two protein sample, that is one is antigen and another one antibody, we mix them together, and when we mix them, we are expecting that they are always forming a full complex like this one. But there is a possibility that there will be some molecules that either antibody or antigen will be non-functional, and they will not form this kind of complex anymore. If it will not form this kind of complex, what will happen? It will be free trimer, you can find out some places, and you can find out this kind of antibody some places at a different orientation. Now if you look at these images, you can see these things clearly. You can see this clearly from those images that there are some trimeric shape is available, those are free trimer. This free trimer, they didn't interact with any antibodies, and also here I pinpointed some small molecules, free molecules, those probably free antibodies are there. So in this particular image, we just mix both the samples together, and we are expecting that it is forming this kind of complex, and definitely it is forming this kind of complex, and this complex we can see at the right side here. We can see this one, this kind of complex, and those complexes are represented here. But same time there are a lot of free antigen and antibodies are there. Those molecules we highlighted either orange circle or yellow circle. So the image wise this is an good image, but our sample preparation wise this is not a very good sample preparation. So there is some problem with the sample preparation. So what we have to do for this kind of sample, either we have to recalculate this concentration of both the antigen and antibody in such a way, so we can mix them in such a way that we can always get the full complex. There should not be any free antibody or Fab fragment or trimer. They should form always complex. Otherwise, when we prepare this kind of sample, best way to do it, you run a ACC purification. ACC purification, it will separate the molecules based on their size. If you have the antibody, consider that antibody molecular weight is 450 kilodalton and this one is 150 kilodalton. So, in this particular case you can see the molecular weight wise, when it will form a complex, it will come at least 600 kilodalton. This is a huge molecular weight. So, now if you purify, actually this antigen and antibody, it will elute somewhere. Okay. So, in this particular case I can say this is, in this particular case this is 150 kilodalton, this is we consider 450 kilodalton. Now, when we have the complete complex, so something like this one, if we try to elute the molecular weight wise, it will elute at beginning, so it will be 600 kilodalton. Then if there is any unreacted antigen and antibody, it will stay, they will elute like that. So, what is happening, if we take this sample, most of the time we will isolate a complex sample and that will be well distributed here. So, in that time we will get rid of this 2 sample from this 2 component from our sample, and that will give us a much better images. Now, come to the next image. Here you can see comparatively much better image where you can see isolated particle, but definitely our stain is not that good, or there are some other problems is there. Either we are, if you just remember the ray diagram of the microscope, what we are doing here, we are always expecting that all this crossover point, it will go through the center. Here now you just consider this is the area we are imaging. So, if this is our optical axis, it will come somewhere here. Now, if there is a little bit shift, if our alignment is not proper, if there is a little bit shift like that, so it will come in this way and it will give us some kind of images like that. So, it is deviated from the optical axis, and then definitely when you will try to image, you have a camera here. So, if you have a crossover point somewhere here, you will get some kind of beam in this particular region to image it within that camera, but rest of the part it will come as a very black area. So, you can see this particular area is much thicker than this particular region. So, there is a one possibility is that beam is not centered properly, or this is the area you have a thick stain and this is the comparatively lighter area. So, from this image, you either, if you get this kind of images, it is better to realign the microscope and center the beam. So, that will help you to get a better image. But this image is definitely a better image than previously whatever image we discussed. Now, come to the next image. The next image, we have this kind of images where you can see so in this particular image you can see there are some large molecules or clustering kind of thing, but same time in both the images, if you look at those images from top to bottom, you can see that there are so many small particles are all over the screen. Here you can see they are very closely packed. So, in this particular image, what happened, there are so many molecules are very close to each other. So, when there is so many molecules are very close to each other, you cannot distinguish their shape properly. So, if you remember previously I told you that for negative staining generally we prefer that 0.1 mg per ml. So previously I discussed this one for negative staining and negative staining sample. Generally we use 0.01 mg per ml concentration, and for cryo I mentioned that 1 to 2 mg per ml it will be better. So this low concentration, if concentration is very high, your protein molecule will stay very closely packed with each other. So here you can see, anyone can consider that this is a very heterogeneous sample, because what I showed you, there are too many particles are very close each other and their shapes are not very good, and same time this sample is overcrowded. That means there are so many protein molecules are within this one frame. So there are many protein molecules are staying within this one frame. So if you just consider, so probably this protein sample is 0.2 mg or 0.3 mg or 0.4 mg, and then we prepared our grid, and that is the reason most of the areas are looking overcrowded. And this kind of images, again, it is not a good image. Why it is not a good image? Because here the protein samples are so close to each other, it is very difficult to distinguish the morphology of the individual protein molecules. They are closely packed, and that is one of the reason we cannot identify them properly. Okay. So what about the next sample? If we just consider, so here I am showing you one sample, and if we just consider the staining of this sample, definitely it is a very good stain, because if you look at that sample anywhere, you can see it is a very clearly we can distinguish all the samples here. You can see the background, this is the white background, and then surroundings area you have a black stain, and the protein samples are embedded everywhere, where you can easily distinguish those protein sample within this black stain area. Some area you can see this kind of large molecules, some area there are many things are clustering together. So here also you can see one black patch here, one there is a very large molecule is, we can see it here. Here you can see another large molecule. Okay, here another large molecule, and then again there are some small molecules here, some small molecule here. So when you look at these small molecules and the large molecule, we cannot distinguish which one is our protein. That okay, this large molecule is my protein, this large molecule or this large molecule or those small molecules. So this kind of protein samples are very bad sample, or this protein itself, it is a very heterogeneous sample. So we try to purify that sample, but it is not homogeneous anymore. If we just consider that SCC profile, when we are talking about that SCC profile, so if we just consider that this is my SCC profile, so lot of time I discuss about that one, that when we purify this protein, we will get this kind of peak, or sometime we will get this kind of peak. And now from here, so molecular weight basis it is going to separate. So when you have a peak something like this, you can see there is no separate peak. So here you will get a different molecular weight protein, here you will get a different molecular weight protein, here you will get a different molecular weight protein. So at different area you have a different types of molecular weight. Or if you have a SCC profile something like that, you are getting a, and then so this tells us that our purification has some problem. This protein is highly heterogeneous, and highly heterogeneous, that is the reason we are getting different shape of the molecule, we are not getting one exact morphology. So if you just consider, those are very large molecules, these three molecules are very large. The other hand, if you look at those molecules are comparatively small molecules. So we do not know from here which one exactly our protein and which protein we are looking for. They have completely different shape. So again this sample has a better negative stain. So stain is distributed properly. From there we can isolate the sample easily, but we cannot tell that this sample is a good sample for further study. So that is one of the drawback of this one. So there is a two possibilities, that either we didn't purify our protein properly. When we purified our protein, we may have a different type of SEC profile, or we are getting different peaks. Last class I also discussed about that sometime after SEC purification we are getting multiple peaks, but we are merging all these fractions together before doing the TEM imaging or cryo imaging. So that means whatever based on the molecular weight we separated the protein sample, we mix them again together and then we are trying to image. So if we are doing mix all the protein together and again we are trying to image same, so we will end up with this kind of sample, this kind of images, and that images will not give us any particular protein shape, that it is a trimer or tetramer or hexamer or pentamer. So it is a mixture of everything. There is another image, you can see it here, the same type of image where we are discussing those different shape of this molecule and different size. Now if we go to the next image, we can see there is another two images there. We are in this particular sample, we are expecting some kind of elongated disc shape molecule. So the molecule, if you just have the molecule like that, and if you take the 2D image of this sample, it should look like this. So the sample is like that, from 2D when you look, you will see only these two side. So if you look at this particular image, what we can see here, so there are lots of sample. If you just look this one, whatever I showed, the similar shape you can see it here. There are some molecules are like that, but there are some very large molecules are also there. In the same image also there are some kind of clustering, or the proteins are coming together or forming this kind of large cluster, or there is a problem with our purification. So in this is our required protein shape, whatever I am drawing with the, if I just use the different color it will be clear. So our required protein shape is this one. The red one is my required protein shape and orange is some kind of aggregation I am showing. So again in this particular image also I am targeting this kind of protein molecules, but actually there is a lot of other contamination is there. Here even you can see a tetrameric sample is also there. So this is a mixture of different types of protein. Overall stain is very nice. Why overall stain is very nice? Because you can see there is a difference between the background and in your protein sample, where these white molecules you can consider as a protein sample and you can see your protein sample very clearly. So that is the reason I am telling this is a good image. If you just consider this is a good image. Whenever we want this kind of negative stain, but if you consider about the sample, this sample is a heterogeneous sample, this sample is a heterogeneous sample. So this kind of sample, when we have this kind of things, that where the different shape, size all are mixed together, this is basically called heterogeneous sample. So when you purify this, if we are getting this kind of pattern, generally it is not a good image for cryo study or any structural study. After your purification you want to cross check your SEC profile peak, that how clean is your protein. For those experiments that is fine, but if you want to use this sample for cryo-EM study or for the structure determination, definitely this is not the good sample or ideal sample for structural study. Now go to the next one. Here I want to show you some lipid protein clustering, because this lipid protein clustering or protein aggregation both are very important. There is a couple of more things we have to understand, that this is a very common problem when you purify our protein, that okay, our protein has different subdomain, or different proteins are coming together and they are interacting. So when we are purifying our protein, we always want that our protein will stay in between 150 millimolar salt to 300 millimolar salt and almost zero glycerol. So generally in presence of glycerol or high salt concentration, protein will be yes, the solubility will increase. So protein solubility will increase that comparatively at high salt concentration and the glycerol, and suddenly drastically if you reduce the salt concentration or if you remove the glycerol completely, so there will be a tendency of the protein that protein will come together and try to cluster between each other. When they will cluster, that is called the aggregation. So there is a basic difference between the oligomerization and the aggregation. Oligomerization, basically proteins, any protomer, it has a particular shape, and the protomers, they will intact their morphology and then it will interact with the another protomer, and then it may form a oligomer, and that oligomer, so if there is a one protomer and there is a one protomer, these two protomer will come together, there is the interface, they will interact and form an oligomer. So in this case two proteins are coming together, interacted and forming a dimer. So in this way it may form trimer, it may form tetramer, pentamer, hexamer, heptamer. So different kind of oligomer is possible. But aggregation is something completely different. Aggregation is something when protein is denatured somehow, or there is a flexible region, or there is a intrinsically disordered region which is not folded, and it is very happy when we have the higher salt concentration or glycerol concentration, and suddenly we remove that high salt concentration, high glycerol concentration, and those flexible region or disordered region, it will be freely accessible within the protein, and then protein will come together and then they will start interacting with each other, and in this particular case they will start aggregating. So that is called protein aggregation. When protein aggregates, it is very difficult to take them back to that original position. So because there is a unfolding is there, and unfolded region, they will try to interact with each other and try to stay together. So mostly that hydrophobic region, if it is accessible, if it is somehow protected by different domain, and if it is open up, so hydrophobic region will try to come closer and the protein aggregation will happen. Here one of that example I am showing here, that okay, the protein aggregation or lipid protein aggregation. So lipid has a hydrophobic region, so that hydrophobic tail is always stay inside and the polar region will stay near the solution. So here you can see there are some images where we can see that protein aggregation in this particular region. If you pay attention, there are proteins are clustering. Here you can see very circular nice protein all over. This circular nice protein, they are isolated from each other. So we are targeting this kind of isolated protein molecules here, but due to presence of lipid and the proteins that hydrophobic region, they are interacting with the lipid molecule in such a way they are forming some kind of cluster. From here you can see one or two isolated protein molecules, but they are clustering in such a way it is very difficult to isolate them from here. So if you see this kind of pattern in your images, so first thing you have to consider, it is completely aggregated. This kind of samples are not good for your any other purposes, but definitely if you do this negative stain is looking very good, it is a very clean image. So, staining wise it is very nice. So, you can isolate the background from your protein sample or aggregation, but overall sample quality is bad. But also from this experiment you can learn something that okay, this particular condition, if I remove glycerol this much or if you remove salt concentration this much, my protein may aggregate, protein may not interact with the this particular lipid, they will just form a cluster. So, from this negative staining experiment you get this information also. So, I will stop it here today, and the next class we will discuss more about the protein aggregation, protein lipid aggregation, and then I will show you some good TEM images. Thank you all for your attention.